Hello, in this video we will discuss about the oxidation of one molecule of glucose. So the oxidation of one molecule of glucose means the biochemical pathway which that is breakdown of the glucose in the intracellular breakdown of the glucose in the into a production of ATP. So let's begin to understand. Here is the starch, basically the carbohydrate containing a polysaccharide, amylose and amylopectin. When we will intake the amylose and amylopectin, which that is semi-digestion in the oral cavity due to the saliva production, while the simple glucose we can also intake. So after the digestion, that is absorption mechanism into the blood vessel, and assimilation. After the assimilation mean the part of the body when the sugar will go inside of the cell. So the blood glucose rise during the fed state. So this is the fed state we will discuss. So the stored in the form of glycogen or consumed as an energy in the cell to produce ATP. So that is two possibility can be. So here the intracellular formation when the move the glucose into the cell. So when glucose will move inside, it means the biochemical pathway will begin. So remember about that. So here is the pancreas, and here is the digestive system as well as the blood vessel. So when the digestive system will digest the complex sugar into the complex carbohydrate in the form of simple sugar, will absorb into the blood by the active transport and on the other hand the pancreas produced by the rising of the glucose blood glucose to stimulate the pancreas to produce insulin from the beta cell, and the glucagon will be inhibit. So this formation is a glucagon will be inhibit to bind with the insulin receptor in the plasma membrane of the cell to stimulate the GLUT4 vesicle. So this GLUT4 vesicle, GLUT4 containing vesicle will be bind, diffuse into the plasma membrane, and after the binding with the plasma membrane, the GLUT transporter. This GLUT transporter basically transport the glucose from the blood into the intracellular cytoplasm. So anyhow, this is the overview of the basically is the glucose transformation from the blood circulatory system into the intracellular part. So remember about that. Here is we will understand the Sympore transport in the liver. GLUT2 channel is used, for example, in the liver, and the sodium will move inside to move the one glucose molecule. It means the water is also carry by the sodium inside, so that is mean the soluble in the water. So the requirement of water for glucose is more. So here is the ATP dependent active transport, which that is also same. So this is the rare. So important thing is that you should understand about here is the glucose will be enter and hexokinase activity to lead used to ATP to lead a production of glucose 6-phosphate. After all, one thing you should remember about that during the fasting and keto diet, it means the production of the we are not eating, and alcohol for example consumption, we are not eating just sugar, so we should preserve the sugar in the form of glycogen still here, and fat will be first consumed to produce ketone. And these if the blood glucose level low, starvation periods, excess of ATP and NADH is come from the basically the ketone and inhibit the glycolysis by the phosphoglucose isomerase to stop. So the phosphoglucose isomerase will be stopped. Why? Because the ketone will produce the energy, as well as for example fasting, the fat will produce energy. So glycolysis no need more to produce energy. Why? Because we are preserving the glycogen first, and first we are consuming the fat, while the fight and flight response produce cortisol hormone, which that is basically also inhibit the phosphoglucose isomerase to produce the ATP from the fat. So remember about that. Sympathetic nervous system stimulate. So here is the phosphofructokinase 1 enzyme use ATP to produce fructose 1,6-bisphosphate. So this was the ATP dependent reaction. It means the energy consuming phase. So remember about that. So during the sympathetic nervous system, we should not use the ATP for consuming ATP consuming process. So it means the glycolysis will be inhibited during the fight and flight response. So on the other hand, the aldolase enzyme, which that is split the two molecule from the glucose, from the fructose 1,6 bisphosphate to produce the glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. So next is the energy harvesting phase. In this phase, the NAD plus convert into the NADH, and basically we are collectively two reaction collage formation we are understanding in this manner. So the phosphoglycerate kinase enzyme used with the water to produce 1,3-bisphosphoglycerate. It means the 1,3-bisphosphoglycerate is basically the two molecule. So this means here is the again 2 ADP used to produce 2 ATP, and the water is used, and the phosphoglycerate kinase enzyme used to produce the kinase means the attachment of the releasing of the phosphate group. So it means the two molecule of 3 phosphoglycerate will be formed. So remember about that 3 phosphoglycerate. After the phosphoglycerate mutase enzyme used to produce the 2 phosphoglycerate, it means the mutation will be occur and carbon number 2 shifting of the phosphate group toward the carbon number 2 rather than the carbon number 3. So that is important. So here is the enolase enzyme, which that is used to add water and remove the phosphate group, and release can be water during the same sympathetic nervous system, which that is lead to backward. So you should remember about that to produce 2 molecule of phosphoenol pyruvate because enolase enzyme and pyruvate kinase enzyme with the using of ADP to produce ATP 2 ATP molecule from produce a 2 pyruvate molecule. So I hope you make sense about that glycolysis. So next we will discuss about that. This is the oxidation of one molecule of glucose. So we need to understand the glycolysis and we need to understand the Krebs cycle. So the glycolysis produce the 4 ATP and 2 U's, and it means the 2 ATP produce and 2 NADH produce without using oxygen. So this is the anaerobic respiration. It means the without oxygen using to produce. So that is the initial process in also in the prokaryotes. So remember about that. But here is the voltage dependent anion channel, which that is allowed to pyruvate move inside due to the voltage changes, and the pyruvate will move inside. For example, we are taking the 1 molecule of pyruvate and we can double this reaction. So remember about that, when the whole product will be double after the pyruvate. Why? Because the glucose molecule is contain a 6 glucose, so 2 pyruvate production. So there is 6 carbon containing compound. So here is when move into the extracellular mitochondrial matrix. Extra mitochondrial matrix, not extracellular mitochondria. And here is the inner membrane, and that is the outer membrane. So when move the pyruvate inner mitochondrial membrane to inner mitochondrial matrix by the Sympore transport. So the hydrogen ion pump inside, the proton pump inside, and the pyruvate will move inside. And after the movement of the inside, the pyruvate will be converted into the acetyl CoA. By this mechanism, NADH will be produced, and the CoA-SH or pyruvate dehydrogenase enzyme used to that and produce the during the CoA-SH to bind with the acetyl CoA to release acetyl CoA, and that is the basically the oxaloacetate is the intermediate molecule in the mitochondria, which that is also here is the carbon dioxide will be produced, that is exhaled during the breathing. So here important thing is that acetyl CoA will be bound with the intermediate molecule for the Krebs cycle in the for electron transport chain use that NADH and FADH2. So here is the cis aconitase or aconitase enzyme used to produce by the binding of the acetyl to convert the CoA will be released and the citrate formation. This citrate is basically contain a 6 carbon containing molecule. This is the first citric acid cycle, so that is known as the citric acid cycle. So the water will be removed or add to produce the isocitrate, which that is reversible and can be reversed. So the isocitrate basically the isocitrate dehydrogenase enzyme used to produce the alpha ketoglutarate. In this reaction, the carbon dioxide one molecule is more released and the NAD plus convert into the NADH. Why? Because the isocitrate dehydrogenase, it means the dehydrogenation of the molecule. So the alpha ketoglutarate is the 4 carbon containing compound which that is after the alpha ketoglutarate dehydrogenase enzyme used to produce NADH. So that is the dehydrogenase enzyme, and CoASH is the coenzyme will be bind and to produce the succinyl CoA, and this succinyl CoA will be converted by the succinyl CoA synthetase enzyme to produce ATP from the GTP from the GDP, and these GTPs after are converted into the ATP from the ADP. So that is the donation of the phosphate group, or GTP can be used for another mechanism. So for basically used for the G couple protein receptor. So here is a succinate will be formed after the succinyl CoA synthetase enzyme to produce succinate, and this succinate is basically converted into the fumarate in the presence of the water, and the FAD will be reduced into the FADH2 and the succinate dehydrogenase enzyme used, and this FADH2 is also succinate dehydrogenase is the part of the electron transport chain. We will discuss in later. We should understand about that, and convert the fumarate and fumarase enzyme used to produce malate, and this malate dehydrogenase enzyme used to produce NADH, and that's produce the oxaloacetate, that is the intermediate, basically is the intermediate, and it can be again the chemical pathway will be occur in the Krebs cycle. So that is the important things you should understand. So that is the basic concept about the oxaloacetate, which that is the intermediate, and this intermediate is basically pyruvate decarboxylation, and here is the pyruvate decarboxylation, and pyruvate decarboxylation to produce acetyl CoA. So that is important things. By the oxaloacetate will be bind with the acetyl CoA to produce again citric acid or citrate. So here is the important thing is that Krebs cycle is contain basically 4 NADH plus 2 pyruvate, it means the 8 NADH, and on the other hand the glycolysis is basically glycolysis produce 2 NADH, while the Krebs cycle produce the 8 NADH, as well as the another thing is that the 2 FADH, and this produce the 1.5 ATP from the FADH, and 2 ATP is the extra. So remember, this is the we should need to understand that the preparatory step is that during the pyruvate conversion into the acetyl CoA, that is the preparatory step, and preparatory step produce the 2 NADH. So that is why. So here the Krebs cycle produces total 6 NADH, NADH, and it means we can classify and we can imagine that. So basically here is the total, basically you should understand about the Krebs cycle. 8 NADH from the 1 glucose, so not 6 but 8, and FADH is 2. Here is the Krebs is not 6 but 8 is produced from the Krebs cycle. So remember about that. Here we are understanding with the calculation to understand the where the NADH is where come from. So here is the glycolysis produce 2 NADH basically in the cytosol, so in the plasma cytoplasm, and 2 pyruvate produce, and 2 pyruvate will produce 2 NADH during the production of acetyl CoA, and this is the preparatory step, and the 2 acetyl CoA will lead to produce the 6 basically 3 NADH, and it means the basically 6 NADH, and basically here is you can see that total basically is the 6 NADH. Why? Because it produces 6 NADH due to the 2 molecule after the preparatory step, and Krebs cycle produce also 2 FADH2 and during the Krebs cycle. So the total 10 NADH, while the 2 FADH2 is produced from that. So that is here is the basically general equation to show that the glucose, which that is broken down in the presence of oxygen anaerobically to produce 6 carbon dioxide molecule and 6 water molecule, as well as the 36 molecule of the glucose, 36 molecule of the ATP. So here is the mitochondria. Again, I am trying to understand that. So basically, mitochondria contain a double membrane organelle. It is known as the powerhouse of the cell to produce ATP. So that is why, and it's contain a inner mitochondrial matrix and outer mitochondrial matrix, and between both is the inner mitochondrial membrane and outer mitochondrial membrane. So contain outer matrix and inner matrix. So that is the mitochondria. It is organelle which that easily to produce the ATP. So here is the outer mitochondrial membrane. So we can imagine that, and this outer mitochondrial membrane contain also enzyme. This enzyme is basically is known as the glycerol 3 phosphate dehydrogenase, is the cytosolic enzyme. So this basically enzyme used for the glycerol 3 phosphate shuttle mechanism. We will discuss later, not here this time, but in this video later we will discuss. So here is the plasma, and here is the basically I want to share that the electron transport is the redox reaction. It means the reducing reaction, and accumulation of hydrogen ions within the matrix. It means the within the matrix the hydrogen ion accumulation and passing through ATP synthase enzyme. So basically the outer mitochondrial matrix will be accumulate the proton, which that is ultimately go inside of the inner mitochondrial matrix by the ATP synthetase channel. So here is the adipose tissue. For example, I want to share that concept. Adipose tissue will be produce the give the LDL fatty acid to the LDL by the lipolysis during the fasting state. So the lipase enzyme here is the blood vessel contain a lipase enzyme, which that is dissociate from the LDL to produce free fatty acid. This free fatty acid can move inside by the channel and to use as a beta oxidation inside of the mitochondria to also produce ATP. But here we will not understand about that. So here is the muscle tissue also produce the glucose due to the glycogenolysis. So the glycogenolysis will produce the sugar, and here is the sugar in the cytoplasm will go into the glycolysis. Glycolysis means the breakdown of the glucose, and to produce 2 NADH is the outer in the cytoplasm. So these 2 NADH will enter in the different mechanism we will discuss also. That is known as the glycerol 3 phosphate shuttle mechanism. So here is the voltage dependent channel, which then move the pyruvate inside in the outer mitochondrial matrix. After the movement of the outer mitochondrial matrix, then the pyruvate will move inside into the inside of the mitochondrial matrix. So while here is the channel, this channel is basically is known as the MPC channel, which contain a 2 subunit, that is the monomer and basically the dimer, and that is pyruvate will be move inside, and the pyruvate in the presence of the pyruvate dehydrogenase enzyme to produce acetyl CoA, which that is later we had discussed about that in the previous diagram to understand the Krebs cycle. So we'll need to go into the Krebs cycle, the acetyl CoA to produce 10 NADH and 2 FADH with the 2 pyruvate. So remember about that. I hope you make sense in this manner. So the electron carrier NADH and FADH2 is the electron carrier. So remember about that. This electron carrier which that give the electron to the inner mitochondrial membrane containing a complex. This complex is made up of protein. So first of all, here is the NADH dehydrogenase complex 1. This complex 1 is also contained with a 45 amino acid chain. This amino acid chain is basically contain iron sulfur protein. This iron sulfur protein and FMN protein is basically give the electron to the complex 1 with the bind with the iron sulfur protein. It means the NAD H will oxidize to produce NAD plus. So here is the next electron channel we will discuss here, that is the 2 complex. So that is the complex 2 of the electron transport chain, which that is known as the succinate dehydrogenase. During the glycolysis, during the Krebs cycle, basically succinate dehydrogenase enzyme used to give the FAD convert into the FADH2. So here is the cytochrome oxidase, which that is used for the oxidation and reduction also. That is the complex 3, oxidation and reduction of the mobile ubiquitinone. And last one is the fourth is known as the cytochrome oxidase, which that is used for the oxidation of the molecule. So let's begin to understand how this process, so how this work. And here is the FN protein, which that accept the electron, while the basically important thing is the iron sulfur containing protein is the central part, which that is accept the electron, and this electron will go into the ubiquinol, which that is 2 proton will be accept. Basically 2 proton is released from pump out in this mechanism, and the Q, which that is ubiquinol, which reduce into the ubiquinone, and this ubiquinone is basically is the reduced into the QH2, which contain a 2 electron and 2 proton. In this manner the 2 proton is more pump out. The total proton is the 4 proton is pump out in this cycle, in this complex. So remember about that. And here is the FADH2 is basically inhibiting this mechanism, while the ubiquinone will go into the complex, passing out the succinate dehydrogenase, which that is not mechanism. And the ubiquinol is a mobile protein. Basically it is lipid soluble, and this lipid soluble can mobile into the inner lipid containing core. So this is basically hydrophobic core, which that is the lipophilic, and this lipophilic core, which that is able to move the mobile ubiquinone. And this mobile ubiquinone is basically inhibitor is the antimycin A, which that is the bacteria secondary metabolites and to produce, and that can be inhibited by this. So remember about that. This is the, for example, the bacterial infection with a specific species of the bacteria can inhibit this electron transport chain. So here is the hydrophobic core here. So when here is after the donation of the electron to the cytochrome oxidase, it means the oxidation, while the also used for the reduction. Why? Because the cytochrome B will be reduced into the cytochrome, and basically here the 2 proton will be accept, and this 2 proton is more pump out. It means in this phase the just only 2 proton will be pump out by this mechanism. Why? Because the loss of energy is too much in this mechanism of the electron, and that's cytochrome C will be reduced. Why? So that is why oxidase enzyme and reductase enzyme used collectively during the QH2 convert into the Q is oxidase enzyme and the reductase enzyme, which that is produced, and basically to produce the 4 proton pump out. So remember about in this manner 4 proton, but 4 complex 4 is pump out 2 proton. Why? Because the loss of energy is too much. So in this phase the cytochrome C carrier of the 2 electron will donate the complex 1. Basically the complex 4 contain 3 subunit of protein 1 2 3, which that is the donation of the 2 electron into the complex monomer of one subunit to pump out the 2 proton. Why pump out 2 proton? Because the loss of energy is too much. So the electron is basically weak with the containing of high energy. So remember about that. Here is the 4 proton with the complex 1, and the complex 3 contain 4 proton, but here is the 2. Why? Because the QH2 is basically containing 2 proton, which that is released already, but the 2 proton is also pumped out due to the high energy. So after this manner, basically here is the ATP synthetase enzyme, which that is the channel-like structure, which contain a subunit, many subunit. For example, here is the proton carrier subunit, which that is contain electron. So the basically is the alpha subunit, epsilon, beta, and gamma. Basically is the upper part is the alpha, and down part is the epsilon and beta subunit, and here is the alpha beta subunit is the downward. So the upper part is known as the F naught, and inner part is the inside is the F1 part, F1 subunit. So basically is the negative charge. So that is attract the, but here is the unstable negative charge. Basically is add the proton and throw by the alpha to enzymatically activity, and the basically beta epsilon to throw the 4 proton to produce the 1 ATP. So this, after all, this electron, where the electron? So basically oxygen, half molecule of oxygen and 2 proton will reduce in the form of H2O, and that means the electron is reduced in the 4th complex. So this 4th complex is the final electron acceptor is the water. So the water is the final electron acceptor. After the water production, basically the that is no need to more chemical reaction again. So that was the azide and carbon monoxide and cyanide, and cytochrome A3 is the cytochrome oxidase inhibitor. So here is the important thing is that the 2-pyruvate is basically produced. Here is the complex 3. We will not here discuss about that. 2-pyruvate will produce the 2 NADH basically, and that is here is the 6 NADH will produce 24 proton as well as. Why? Because calculation is done and total will be 60. So in the end of the video we will calculate again. So here is the tricyclic acid, basically citric acid cycle, which that contain a succinate molecule. This molecule is basically look like this manner, and this work is like that. Basically the succinate will go into a basic part is the succinate dehydrogenase enzyme are present in the electron transport chain, and it contain A, B, C, D is the subunit. Basically it is also contain iron sulfur protein, and in this manner this enzyme is basically FADH2 will FAD plus will convert into the FADH2, and after this conversion the succinate dehydrogenase enzyme convert the succinate into the fumarate, and this fumarate will be again go into the Krebs cycle. But important thing is that you should need to understand the FADH, which that is not involved in the complex 1. So remember about that. So that means important thing is that the total proton pumping through the FADH2 is the 4 is the complex 3 and the 2 is the complex 1. Complex 4. So it means the total is known as the 6 from the 1 and FADH2. So here is the outer side of the cytoplasm. Basically here is the glycerol 3-phosphate dehydrogenase enzyme, that is the cytosolic enzyme, and the 2 NADH will be moved by this mechanism to convert by in the presence of dihydroxyacetone phosphate to convert glycerol 3-phosphate. If you remember about in the glycolysis, basically that is the step of the glycolysis to produce again the 2 NADH, and this 2 NADH is basically is conversion, and after the again conversion of that is produced the glycerol 3-phosphate. This glycerol 3-phosphate will go into the glycerol 3-phosphate dehydrogenase enzyme is here, and after the binding with the glycerol 3-phosphate dehydrogenase enzyme to produce the FAD plus to convert FADH2. So in this manner basically Also 6 proton will be produced from the FADH2 from the NADH. So that is known as the glycerol 3 phosphate shuttle mechanism. So important thing is that you should understand about that. From the glycolysis, the NADH will be produced the 6 proton, while from the Krebs cycle the NADH will produce the total 8 proton. Total sorry, 4 and 4 8 and 10 proton. So that is why. So here is the inhibitor is the basically the succinate dehydrogenase inhibitor is known as the TTFA. That is the inhibitor and stop the electron transport chain. So next one is that important thing is that the glycolysis produce the 2 NADH, and it means the 8 NADH is collectively 8 proton is collectively from the 3 complex, and the 4 complex collectively is the 4, and total ATP is the 3 ATP divided by 12 divided by 4 and 3 ATP, and the 2 ATP is the extras. It means the total 5, and here is that 30 ATP is produced in this mechanism. 32, you can say that 32. So here is the complex 1 is basically inhibited by the rotenone and amabarbitol, as well as amabarbitol, and on the other hand the pyridine, and that is reduce the ATP and the consumption of the oxygen also. So here the 10 NADH will be produced, and the 2 FADH2 is produced from the pyruvate. So basically is the FADH2 is the bypassing from complex 1. So the complex 1 is not involving in this mechanism. So thing is that the here is the acidic medium will be produced due to the increasing of the proton, which there is the final electron acceptor. The water will be reduced the acidic medium, and on the other hand the electron is also reduced. On the other hand, the 4 proton is used to produce 1 ATP. So it means the 10 proton is produced the 2.5 ATP, and FADH2 produce the 10 proton. It means and 4 is the negative. So it's 6 proton is produced, and 6 proton produce 1.5 ATP. So the total you can calculate. You can check that graph. And energy is produced from the ATP used for the muscle contraction, heat production, and homeostasis, etc., etc. So here is the proton cycle. This proton cycle is produced, and basically this proton cycle is produced a proton, and a gain cycle will become. So thanks for watching. I hope you make sense about this video. If you like this video, please comment and like.